you express my gratitude to all of you for being here, and I sincerely hope that you will go back with a replenished mind to understand what next generation sequencing is all about, why we do next generation sequencing, and why it's important for you to learn some of the methods of analyzing data that comes out of a next generation sequencing. I have been told that the audience that I'm facing today is very heterogeneous, not just in terms of their seniority. There are faculty members here, there are also undergraduate students here, I'm told. Also, not all of you are exposed to the field of genomics, but obviously I have to make some assumptions. I will assume that all of you are from a biology background and therefore know what DNA, genes, and so on and so forth. That assumption I'm going to have to make. Other than that, probably I will not make any assumptions. And because I'm not making any assumptions, there are certain portions of my talk that will be too rudimentary for some of you. And there will be some areas, some portions of my talk that will go very tangential to you, primarily because you don't have the background to be able to understand necessarily those kinds of points that I will make. So what I'm hoping is that from my talk, each one of you will be able to learn something about next generation sequencing. So the way that I have titled my talk is next generation sequencing, a technology that has allowed us to ask and answer important questions. Also, I want to start with a disclaimer that I actually may not be able to end my talk because I am going to go slow like a teacher. It's not my duty to just finish my slides. That's not my duty. My duty is to be able to relate to all of you, and therefore I may not be able to finish all my slides, but that really does not matter to me at all. So what I really wanted to impress upon you is that this is a technology. Next generation sequencing is a technology. And why are we attempting to adopt this technology? The reason why we are attempting to adopt this technology in my own mind, and I'm going to give you just one example to drive home the point. The examples can come from different kinds of fields, but if I start giving you examples, then I won't be able to get to the meat of my talk. So I will give you just one example, that this is a technology that has allowed us to ask and answer important questions. Does that mean that we never asked important questions in science? Of course we did. But there are certain kinds of questions, and like I said, I'm going to give you one example. There are certain kinds of questions that we asked and we weren't able to answer because answering those questions needed appropriate technologies, and those technologies were not available. And next generation sequencing, or massively parallel sequencing to be technically correct, that's what it is called, massively parallel sequencing, is one such technology which, before the advent of which, we were thinking of asking these questions, but none of us had the wherewithal to be able to answer the questions, primarily because we couldn't generate the data and we wouldn't be able to tackle those problems without data generation and analysis. So next generation sequencing is a new technology, new meaning it's about 20 years old now, maybe 25 years old now. It's become very popular in the last 10 years or so, and so that's what I'm going to describe to you, and that's what this workshop is all about. The workshop is trying to promote this new technology primarily because it empowers you to ask certain kinds of questions that you wouldn't dare to ask if this technology was not available. Incidentally, if I'm speaking too fast, you have to slow me down. Alright? I don't want to lose any of you. If I'm not being clear, just raise your hand and I will repeat. Yeah, it's not like an inaugural talk that one gives without relating to the audience. That's not my job. That's not what I'm going to do. Yeah, so if I'm not being clear, please, you're welcome to raise your hand and I'm happy to repeat that. Like I said, that I do not have an agenda, that I have to finish that many slides. No, I don't have that agenda, yeah. So please do not hesitate to stop me as I'm talking. in an inaugural talk, usually people don't interrupt, people don't ask questions, but I'm giving you that permission to interrupt and ask me questions. I don't really want to be vague to all of you. Alright, so the example that I'm going to give you is from one area that I have been working on for the last 10 or 12 years. There were certain kinds of questions that we were asking, and we were not empowered to be able to answer those questions even if we did ask those questions. We now know, and people have been with this idea for the last 50 years or so, that cancer is a disease of the genome, meaning that cancer does not happen unless certain genetic alterations take place. So cancer is a disease of the genome. So if you talk about a disease of the genome, if you talk about a genetic disease, one of the hallmarks of genetic disease is that it will run in families, right? Mendel had taught us about dominance, recessivity, and so on and so forth, so it will run in families. So we will inherit some genes from our parents, and therefore we will be at risk, and at some point of time develop the disease and so on and so forth. So every generation will have certain number of affected individuals. If you look at cancers, and I'm pretty sure that each one of us has encountered a relative with cancer, right? And if you thought about it, if you look at that relative and ask, were the parents of this relative, did the parents of these relatives have cancer? The answer is usually no, right? It happens in one individual, probably to skip certain number of generations, some number of generations ahead of this individual, somebody had cancer, etc. But that's not the hallmark of a genetic disease. A genetic disease we learned about 1-to-1 ratios and so on and so forth. Mendel taught us different kinds of ratios, etc. It's usually familial, but in cancer it does not happen. In cancer, it's really not a hereditary disease. Only about 20 to 25% of cancers, or even less, 10% of cancers are familial. So in about 10% of individuals with cancer, you can trace a relative, an immediate relative in the preceding generation who may have had cancer. And therefore, it runs in the family, and therefore we call it a familial cancer. Most cancers are not familial. 90% of cancers are not familial. This individual has cancer and dies, but does not pass on the genes to the next generation. How does this happen? Mr. Mendel could not even envisage, could not even foresee that such are the kinds of discoveries that will be made, such are the kinds of questions that will be asked. So he only thought about, by the way, I'm not denigrating him, without him we wouldn't exist today, right? Our understanding would not exist. So I'm not really trying to denigrate him. He only talked about those kinds of traits or changes in the DNA that are passed on from the parents to the offspring. Yeah, so those changes are also become heritable changes, and some of these heritable changes predispose us to certain kinds of diseases. But here we are talking about genetic changes that are not heritable, and that happens if these changes do not take place in the germline. Germline changes are inherited, are transmitted from parents to offspring. Non-germline changes are not transmitted, right? They are not inherited. They happen in that individual. If it has some impact, if those non-germline changes had some impact on the individual will suffer or will gain some specific kinds of traits and certainly will not pass them on. Those are called somatic alterations, right? You have heard these words, germline alterations and somatic alterations. I'm being like a school teacher primarily because I'm told that there are undergraduate students here as well. So you will have to bear with me. Like I said, that I want everybody to understand the basics of what I'm talking about. So please bear with me if I'm talking in too simple terms. So these are because these individual cancers are mostly because of somatic mutations. They are non-familial. Yet cancer is a genetic disease because somatic mutations are taking place in the DNA. So cancer is a genetic disease. It's a non-hereditary disease. Only about 10% of cancers are hereditary, the remaining are non-familial or non-hereditary. So the question that we had always asked is, how can we find out whether a cancer patient who is in whose family cancer is not running in the family, how can we find out whether what kind of genomic alterations are taking place in this individual? We had asked these questions, my predecessors had asked these questions maybe 80 years ago, and of course they had no means of answering this question. The means of answering this question came about when new technology platforms came. New technology platforms that allow us to catalog somatic mutations, and until that time we wouldn't be able to do it. How were we cataloging genetic mutations? Of course, I imagine that most of you know what polymerase chain reaction is. You probably also know what Sanger sequencer is. I just visited the Sanger sequencing facility here, so I'm hoping that sometime during the next 2 days, today or tomorrow, you will be able to see a Sanger sequencer if you haven't seen any. But again, these are unhelpful for cataloging somatic mutations, and let me explain why they are unhelpful. Well, I actually want to tell you that cancer is an evolutionary disease. Evolutionary in the sense that these genomic alterations, these alterations in the genome, are taking place over a period of time. And these changes actually evolve to produce this disease called cancer. So I'm going to give you an example. So imagine that that green line is the epithelial layer. So this is like the epithelial layer, and these are cells below the epithelial layer. So when we are born, every cell looks exactly the same, and they are all stacked well beneath the epithelial layer. This need not necessarily be the epithelium, it can be another organ, it can be the lining of another organ, etc. But this is usually what happens, that when one is born, every cell looks the same. Then as we move along, we are exposing ourselves to air pollution. Some of us are probably smoking, and therefore our lung cells are being exposed to certain kinds of carcinogens and mutagens. Given all of this, mutations start taking place. Mutations are taking place in these cells, in the cells of the lung, in the cells of the skin, and so on and so forth. And please remember that these changes will not be passed on to the next generation, will not be passed on to the offspring. These are true somatic mutations. So as we move along, somatic mutations take place. So these are mutations that are happening in those cells. Now those mutations cause us no harm. We are completely oblivious to the happening of those mutations, and so this happens in the background. And this doesn't happen in all cells, it takes place in certain specific cells. These are somatic mutations which will not be passed on. Alright? So more mutations happen, there are more mutations, and these mutations are happening all the time. As I'm talking, as you are sitting there listening to me, we are exposing ourselves to these rays of the tube lights and so on and so forth, some of which can cause mutations, and so these somatic mutations are happening in the background in all of us. Sometimes these somatic mutations cause certain funny things, and those funny things sometimes also cause diseases. So one of the funny things that happens is that, for example, this second mutation that took place gave this cell some growth advantage, as a result of which that particular cell made photocopies of itself, made a clonal propagation of itself. Instead of 2 to 4 to and so on and so forth, 2 to the power of n cell division, this cell suddenly made a number of photocopies of itself called clonal propagation. This is not normal cell division, it's an abnormal cell division. That abnormal cell division was provided by the second mutation. So that mutation gave the cell a growth advantage for it to become a small clone. Again, these small clones are happening all the time in our organs. We don't care about them because they cause us no bodily harm, but those mutations are providing the cells a growth advantage. Again, another mutation takes place in the cell, and this mutation provides the cell a little bit more growth advantage, so that we have a larger clone of cells compared to this clone of cells. But again, this larger clone of cells is not causing any harm to us, So we don't care about them. Even if we get to know about them, we don't care about them, and we usually don't even get to know them. And this process continues. So here is another mutation that has taken place, this red mutation. This mutation has taken place in a cell that already had two pre-existing mutations. Those two pre-existing mutations caused no damage to us. So that we don't expect this new mutation, the red mutation, to even cause damage. Unfortunately, the red mutation gave this cell a huge growth advantage in that the cell breached the epithelial layer and became a malignant tumor. This is exactly how most cancers take place, most non-familial cancers take place. They are because of somatic mutations. So what is our job? We need to identify this red mutation, which mutations are providing the cell with a huge growth advantage such that it becomes a malignant tumor. And we need to then be able to identify this mutation and get it back onto the genome of the individual, in which gene is this mutation taking place or has taken place. And what does that gene do? What is the kind of alteration in the function of that gene because of the mutation? Because if we know function, then we know how that particular mutation gave the cell a huge growth advantage and it became a malignant tumor. So that's what we need to do. But as you can imagine, and even after this red mutation happens, the same process goes on. There are mutations that are happening in the background, right? So lots of mutations are taking place, and the question that we ask is, what are the genes and genomic alterations that drive? It drives the cell, it provides the cell with a huge growth advantage, and because of that it becomes a malignant tumor. So our task is to be able to identify those changes, somatic changes, and the functions of those somatic changes. Might sound very easy, but it's a hugely difficult task. Why is it hugely difficult? To be able to identify that red mutation, which actually leads to this malignant tumor, is not going to be easy because in the background there are so many other mutations that are happening. So we have a lot of background noise, and this red mutation is like the signal. To be able to fish out the signal from this huge amount of background noise is not an easy task. It requires a lot of computation. First of all, data generation. It requires a lot of computation. It requires a lot of bioinformatics knowledge, because you now want to take this mutation, put it back on the genome, identify in which gene this mutation has taken place, and then what does that gene do, and why does it promote the growth advantage to the cell. Right? It's a huge set of one after another questions that we ask. These are not new questions. These questions, like I said, had struck my predecessors, and there was no way that my predecessors could actually even attempt to answer these questions. Why were they not able to answer these questions? Well, let me, by the way, we must remember, as we are talking about next generation sequencing, we must remember this gentleman who was a Nobel laureate. He wrote a paper in 1986 in the journal called Science, and this paper was prophetic. What is the prophecy that he made? What was the prophecy that he made? And I'll read out just one sentence from this particular paper that has become a classic now. A turning point in cancer research is the name of the paper, and it had a subtitle called Sequencing the Human Genome. What did he say? He said that if we wish to learn about cancer, we must concentrate on the cellular genome. We have two options: either to try to discover the genes important in malignancy by a piecemeal approach. The piecemeal approach is what we all did most of our lives. My predecessors did throughout their lives, which is to look at certain genes called candidate genes and try and find out whether there were genetic alterations in those candidate genes. They did not have the wherewithal to look at the genome in its entirety, the 3 billion nucleotides that we have in its entirety, primarily because we did not have the platforms, the machines to be able to sequence. But he emphasized that it is absolutely critical to do this, and the reason why he emphasized that is that this piecemeal approach will only teach us piecemeal basis of cancers. If you want to learn more about cancer, we need to sequence the whole genome. It will be far more useful to begin by sequencing the cellular genome. That's what he said. He said genome sequencing is something that's absolutely critical for us to be able to learn more about cancer. And I've just given you an example as to why these somatic mutations are important, and because they give us this growth advantage. But why is it that we couldn't actually identify somatic mutations given the kind of technologies that we had? And you will see the technology that we had in the form of Sanger sequencing, PCR amplify, and then use the Sanger sequencer to sequence that particular fragment and so on and so forth. And the reason why we won't be able to do that is the following. So the question is, why couldn't we catalog somatic mutations using a Sanger sequencer? Sanger sequencer, of course, relies on amplification of portions of the genome, and the way that we do it is that we design primers, and based on these primers, the DNA piece will get amplified. Then we go and take that amplified piece and sequence it, so we get the nucleotide sequence of that amplified piece, right? Everybody knows this, that's what PCR does, and that's what DNA does. I'm going to tell you why we can't rely on Sanger sequencing technology to be able to do that. So imagine that these are all pieces of DNA that we are going to amplify using, and then Sanger sequence. And let's say that in these, I don't know how many DNA pieces are there, but whatever, some number of DNA pieces, and these DNA pieces have this particular mutation, and these DNA fragments have that mutation. And what is it that we want to do? We want to capture both mutations, right? Ultimately, we want to sequence, and we want to say that there are two mutations in this set of DNA sequences. Now, these DNA sequences are coming from one individual. Different cells are producing, different cells have different kinds of DNA sequences. These are all somatic mutations. Now, if you design primers from this end and this end and try to PCR amplify, what you're going to get is, as you know, through the cycles of the PCR, and if you don't know it, I can't explain it right now because it's going to take me a lot of time. Through the different cycles, the DNA sequence that has the most prevalent mutation, that's the one that's going to be eventually come in your ultimate cycle, whatever, the number of cycles. So what you're going to do is what you're going to get after the PCR is just this set of DNA fragments. These will be lost. You will not get those. So ultimately, after the completion of your PCR cycles, these are the DNA fragments that will remain, and what you are going to be able to catalog is this most frequent mutation in this set of DNA. The minor mutation is just going to be lost as a result of the PCR. Therefore, we aren't, if you look at, and I can plug in more mutations, and you will see that you will not be able to identify, catalog all the mutations present in the set of the DNA. And that's the nature of the beast. That's how PCRs happen. How then can we identify both mutations? Well, one way to do this is very simple. If we can, instead of using PCR primers here and here, supposing we fragment it here, supposing we cut these DNA pieces here and separately PCR amplify these, then what we're going to see is that in that first fragment, this is cut here, and if you place all of these into the PCR machine, what you're going to get is this particular amplicon containing that particular mutation. What about the remaining? We have cut this, and this is another PCR sequencing that you're going to do, and there you're going to get this particular mutation. Now, if you put both of them together, you have now captured both the mutations. You couldn't do it if you did it in one go. This is essentially the principle that next generation sequencers use. So on the basis of this is what you're going to get if you amplify them together, but this is what you're going to get if you have done it separately. So the idea is to be able to cut it here and then sequence both. But I'm able to cut it here because I've put the picture that these are the two mutations. How do we know where to cut when you don't even know where to cut? So what we do is we say that, well, we'll just take a chance. We will fragment the DNAs randomly. So there will be some where one particular mutation will be caught. There will be some fragments where the other mutation will be caught. So as we sequence all of these fragments, we hope that we will be able to capture all of the mutations and catalog all of them together. That's essentially the basis of the next generation sequencing or massively parallel sequencing. Now, if you're going to do it one at a time, it's going to take a hell of a lot of time. We probably will spend our lifetime sequencing one genome, but this is done in a very parallel way. I don't know if you have in this workshop, if somebody is going to teach about the basis of massively parallel sequencing, the parallelization that takes place in the technology platform. If you're going to teach that, then do emphasize how we are saving on time by parallelization. Otherwise, I mean, if you're going to do it sequentially, one after another, it's just going to take our whole lifetimes to be able to catalog all mutations. So the next generation sequencers are called massively parallel sequencers, but this is essentially the principle that underlies next generation sequencing. So what we do is we randomly fragment. This is the DNA sequence. We just break the genome into small pieces called fragments. So we randomly fragment it, and then we prepare a library, and essentially then we assemble or we sequence each of these small fragments. Many of them will be overlapping, as you can see, because they are randomly fragmented, right? So there's an overlap between this fragment and this fragment, there's an overlap between that. So we then sequence everything and then try and figure out where the overlaps are. So I'll tell you a number of problems that arise because of this kind of random fragmentation and sequencing small bits and pieces of the genome. After all, this is my genome, this is what we want to reconstruct or capture, but then we are fragmenting it and we're only getting bits and pieces. So how do you know which piece comes before which piece and which piece comes after which piece and so on and so forth? So how do you put them together? How do you put the sequences of all of these n reads, one of these fragments, when it's sequenced, it's called a read. So how do you assemble all of these reads into one single strand, which is what you started with? That's the big challenge. This is now completely standardized. So there are technicians who will actually fragment the DNA, who will prepare the library, who will sequence it and generate the data and give you the data. So that's been completely standardized. What is not yet standardized is, well, much of it has been standardized now, but still there's a lot of research that's going on. What is not completely standardized is how do you put all of these things together to reconstruct this one single DNA strand. Okay, and that's what this workshop will be all about. This workshop will emphasize how do you put them together and make that reconstruct that single strand. After all, it's a long rope, and you have broken that rope into multiple pieces. You have to reconstruct that rope, and that's what requires a lot of imagination, a lot of techniques, and so on and so forth. That's what is going to be done in this. I will give you a little bit of hint as to what we do. And as a matter of fact, the hint is here. What we do is we use these overlapping fragments in order to construct longer fragments. Because of this overlap, we know which one comes after which. So we get longer pieces, and then we take these longer pieces and put it together and against the genome, and then we try and reconstruct that entire genome later. There's also another question that I'm not going to address, but will be addressed in this workshop. Having reconstructed this genome and having identified that those somatic mutations are in specific positions of the genome, how do you identify to which gene that change belongs? And what does that gene do in terms of function? So maybe that gene is involved, or maybe that the product of that gene and multiple, if it's a monomer, then it's just that one gene that's making up a protein which is folding in three-dimensional space, and mutation in that gene might actually screw up the three-dimensional configuration of that particular protein. If it is a multimeric protein, then the problem becomes even more difficult. So these kinds of questions are also require a lot of modeling, bioinformatic modeling, computer modeling, to be able to reconstruct the the original protein structure is known. What you really are asking is that these mutations, these genomic alterations that are taking place, how is it changing the structure? I'm not going to deal with that, but that will be dealt with in this workshop. So massively, when it began, it was very, very expensive. And the expenses have, if you look at linear change in expenses, it would have been like that, but from about 2007, 2008, the cost of sequencing began to fall. Again, I'm not going to get into the reasons why it began to fall, but as you can see that instead of linear decrease in cost, there was a sudden exponential decrease in cost, as a result of which these next generation sequencing technologies became accessible to all of us. If you walk over to the National Institute of Biomedical Genomics, you will see that there are two sequencers that can sequence, I don't know how many genomes a week, lots of genomes a week. The first sequencing of the human genome took about 13 years. Now we can sequence all of our genomes in about a month. So that's what the technology platforms are. Its speed has increased, parallelization has increased, and cost has fallen, which is the reason why we can legitimately discuss about this, because we can bring it to our lab and start generating data and analyzing data. I'll skip this. So one of the first uses in India of the next generation sequencing, large-scale uses of next generation sequencing, was in the International Cancer Genome Consortium project. This particular project is the most ambitious biomedical research effort since the Human Genome Project. The Human Genome Project sequenced the first human genome. And then we started looking at cancers, and a consortium, a Cancer Genome Consortium was formed, and we all participated in this, and we looked at 75 different types of cancers. When I say we, India also participated in this, and we participated with, so you can see that the Indian flag is shown there. We participated with oral cancer, and this project, which cost us crores of rupees, in spite of falling costs, was led by the National Institute of Biomedical Genomics. I was the principal investigator, and the clinical principal investigator was Dr. Rajeev Sareen of the Tata Memorial Hospital in Mumbai. So there were two institutions that generated this data, and that's what we did. So that's one, and essentially I have told you why we undertook this project, primarily because we knew that cancer is predominantly because of somatic mutations, and we couldn't touch somatic mutations because the platform wasn't there. But then there were also, and when we started doing the next generation, when we started doing the Cancer Genome Consortium, these next generation sequencers came of great use, without which we wouldn't be able to do the project. There were other kinds of projects that also were happening as a result of these improvements in next generation sequencing. The other project is called the ENCODE project, which is the encyclopedia of the DNA elements, which essentially annotates different portions of the genome by its function. So they are called DNA elements, and we want to know the functions of these DNA elements, and that's what the ENCODE project was all about. I'll skip this, and it used a lot of gene expression as opposed to just DNA sequence. It used RNA sequencing in order to look at gene expression levels, because after all, the function of the gene will involve its expression, and what it does, what that particular transcript does in the human body or human physiology, is what it's all about. So these two projects, the ENCODE project and the GEO project, Gene Expression Omnibus, worked closely together to be able to annotate the function, the changes and the function in the human genome. Then there was another project that's called the International Genome Sample Project, which is also known as 1,000 Genomes Project. Now it's become 10,000 Genomes Project, essentially to look at variation, natural variation in genomes not necessarily related to health, to disease, but natural variation in human genomes that comprise all of us. If we sequence our genome, there will be differences in our genomes because our looks, our behavior, some of it is at least controlled by our genomes, and therefore the reason why we look different, the reason why we behave differently in a specific context is because of these differences that we intrinsically have in our genomes. So this 1,000 genomes project, or now 10,000 genomes project, actually attempts to capture natural variation in human human DNA. And of course, when we look at all of this, we are generating data. Like I said, data generation has been completely standardized, and when we generate data, these generated data need to be analyzed, and simultaneously with the generation of data came the development of bioinformatic methods. Many of them have now been encapsulated in various kinds of software packages, some of which will be described today and tomorrow. And these encapsulated software packages many of them are actually freely available from the UCSC Genome Browser, from University of California, Southern California Genome Browser, and you can use many of those online or even download some of them from your server. There are lots of different other projects that are going on. So people are now talking about personal omics. That is, we want to understand ahead of time, even before, maybe at birth, we want to know, what kind of diseases that we will come down with, will succumb to during our lives, what kind of behavior patterns will we have. So many of these are actually called personal omics. They involve sequencing the DNA. They also involve sequencing the RNA of that individual. And the RNA is definitely more dynamic in the sense that changing the environment will change the gene expression levels or expression levels of different genes. So it's more dynamic in some ways, but that dynamic nature actually provides us with the changes, the differences of our behavior, of our phenotypes. And so all of this is happening, and all of this can happen because of this new technology that's massively parallel called next generation sequencing technology. I'm going to skip all of this. So I mentioned this, that we are fragmenting the DNA piece into small fragments, and we are sequencing them. The first thing to do would be to look at these little fragments, find out which ones overlap, and that will involve comparison of these two DNA sequences, base by base, nucleotide by nucleotide, to figure out where there is an overlap. And then what you do is, once you've identified that there are overlapping sequences, so if two sequences overlap, you know that one is on the side of the other. Otherwise, they could not have overlapped. So you now do a lot of work, do a lot of computational work to be able to identify and order those in some way. And this ordering eventually is called assembly. Eventually will lead to longer reads, and then you try and put these longer reads on the genome. So these longer reads also will overlap, and therefore you know now that this is the left, this is in the middle, because this overlaps with that, and this portion overlaps with this. So you know that this is the only portion that's non-overlapping. This will come in the middle, and this comes on the other side. So you now have a long piece of DNA after having assembled that, but the assembly itself is a big chore. You really need to work hard to be able to assemble those. So this is how it looks on the sequencer. So you're going to get these little fragments and these little pieces. There are some that are so small that it's not even worth sequencing, like this one. It's so small that it's not even worth sequencing. So you put a threshold, and throughout all of those sequences, throughout all of those pieces that are below a certain number of nucleotides. So usually we take between 150 to 100 to 150. That has now increased to about 200 to 250. And the longer the pieces are, the easier it is for you to assemble. So these technologies are also changing. And so then what we do is sequentially we align the sequences. So this sequence alignment is a big task in bioinformatics. So you have sequenced these small fragments, you have to align them, you have to find overlaps, and you have to order them to be able to, from left to right, to be able to reconstruct this original DNA sequence that we are trying to reconstruct. And so this is sequence alignment is an algorithm, or there are multiple algorithms for sequence alignment. That's a part of the toolkit of bioinformatics to which you will be exposed. Then what we need to do is deal with non-unique mapping, in the sense that some of these pieces may not even map back to this particular DNA fragment. Where did those pieces come from? You need to ask yourself and find an answer to that. And then, like I said, that not everybody is going to be, is going to have exactly the same genomic sequence, because we behave differently, we look different, we succumb to different kinds of diseases, and so on. So there are changes in the genomes, and we want to look for these allelic differences, because that's where lies the information as to why a particular disease is caused, if it is a somatic disease such as cancer. So we do want to look for these allelic differences, but this looking for allelic differences does not happen until we are able to align the sequences, and sequence alignment is a big task in bioinformatics. I have multiple algorithms for sequence alignment. The one that is commonly used is this particular alignment that was published in 2001 in this journal called Genome Research. It's a fast search. So you have these pieces, right? You have hundreds of thousands of pieces of that original DNA that comes in the form of a library. You have sequenced all of those pieces. Now, if you take the first piece and ask yourself, to which of the remaining 20,000 pieces does this align to, or does this overlap with? And then you take the second piece and ask the same question. Take the third piece and ask the same question. You're going to spend the rest of your life trying to align. So it needs to be done in a parallel fashion. You take the first piece and you take the 29th piece simultaneously, and you take the 37th piece, you take the 179th piece simultaneously. Ask this question. To be able to do that, you need searching algorithms that are very fast. Otherwise, just the process of alignment takes a lot of time. And this algorithm is something that is innovative. There are multiple such algorithms. One algorithm works better than the other in terms of time, better than the other in terms of its ability to put together correctly, and so on and so forth. So, but this is one algorithm that's quite frequently used. Now, I don't know how much of depth you're going to get into. Like I said, that there are also other kinds of algorithms. So this is a fast search method. There are also other algorithms for next generation sequencing data. This is from a paper that was published in 2010. These algorithms are improving both in terms of its accuracy, improving both in terms of the time it takes to align or assemble. So these algorithms essentially depend on a method called hashing. Again, don't have the time to explain what hashing is all about, but these hashing algorithms actually enable massive parallelization of processing of these small fragments of DNA, and your ability to put together, assemble them together into one large fragment. So these hashing algorithms are a part, were invented by computer scientists, and they're now profitably used by genome scientists in order to assemble small fragment genomic data. Again, I just want to give you an exposure as to the different kinds of things that we do. I don't have the time to be able to explain in detail what a hashing algorithm is and how does it parallelize, but it does parallelize. So, like I said, alignment of reads to the reference genome. So we have the human reference genome that took 13 years to sequence, but then we want to align it back to that genome, the different fragments that you're making. I'm not going to talk about this because you can even assemble a de novo sequence, even if you don't have a reference sequence, which for the human, we are lucky that we have, but sometimes, for a different plant, you may not actually have a reference sequence. So where do you align with? So you have to, this is called de novo sequence assembly. You can start from scratch and assemble them together in order to form a sequence, in order to form a larger fragment, larger sequence without the ability to align. So you just assemble them using de novo algorithms and be able to get to a larger fragment. Again, these are specifics, there are specific algorithms to be able to do that. So the difference between these two is that in one case we have a reference sequence, like the human reference sequence. In the other case, we are trying to accomplish the same task without the luxury of having a reference genome. Yeah? And today there are lots of such problems that are being addressed in terms of de novo sequencing. As we are generating data, these massively parallel DNA sequencers are also error prone. When in fact it should be a C nucleotide, it incorporates a T nucleotide. So you get errors in the sequence data. So you need to quality control, and because unless you quality control, you are going to make wrong inferences about the final assembled sequence, right? So the quality control and base calling, these are two very related issues. And again, for quality calling, because you're simultaneously sequencing a large number of fragments and a large number of DNA sequences, you can actually figure out whether a particular fragment where at a particular place which has been called as a T, in the overlapping fragments they're all called C. Then you know that this particular T call is a wrong call. So that's how, again, quality control algorithms are also bioinformatics algorithms, and therefore we need to understand quality and devise some particular methods of quality control, and that quality control will impact on what is the base that you call for at that particular position. Like I said, not all of us at a particular position will have the same nucleotide. Differences in nucleotide can happen because of poor quality control, which we now hope that we have taken care of. If we sequence all our genomes at a particular position, not all of us will have the same nucleotide legitimately. So that's called polymorphism. That particular site, DNA site, is called a polymorphic site because not all of us will have the same nucleotide. As a matter of fact, on an average, we have 3 billion nucleotides in our genome. And if we compare, if we sequence all of us and compare, what we will find is about 0.1% of our DNA sequences are different amongst us. 0.1% might really seem like a very small amount, but 0.1% is like 3 million. 0.1% of 3 billion nucleotides is 3 million nucleotides. So we are different on an average, any two of us will be different on an average at 3 million nucleotide sites, and that's a lot of polymorphism, a lot of difference. Ultimately, like I said, that I'm not going to deal with it, not even competent enough to deal with these modeling and three-dimensional structures. More falls in the area of biophysics and other kinds of computational biology. So genome browsing and annotation of these changes of these polymorphisms that we detect, etc., also fall in the area of bioinformatics, and we need to deal with it. As we generate this data, Anshman, will you tell me when to stop?
We have some time, sir.
So whenever you are irritated with me, just raise your hand.
I think that no one is irritated.
I will stop. No, it can get heavy, right? So it can get heavy because I'm pumping in a lot of new concepts.
So if you have any question, you can ask us. So
yeah, so I can take a pause right now. If you have any questions, I'm happy to answer. The next set of things that I'm going to do is going to sort of start another new topic, but or not a topic, but yeah, shift of gear. But so I'm happy to answer any questions that you may have.
So on what I've talked about. So you can ask.
So and language might not be a barrier. So if you want to ask
you
me,
can ask in mother tongue. So no problem. No problem.
Yeah. So
don't hesitate.
Go ahead.
Hello sir, I am Shadab.
Sit down. Sit down.
I am from Shadab from Brentford University. So my question is, sir, what do you about say polymorphism? Sir, can you repeat this?
Polymorphism?
Yes, sir.
Okay. So if you sit down. if we sequence all our genomes, each one of those sequences will be roughly 3 billion nucleotides long. If you take a particular position, let's say it's numbered as 1, 2, 3, 4, so on up to 3 billion. Although it's not a contiguous stretch because our DNA is fragmented into different chromosomes, right? But let's say that we have put all of the DNA in the chromosomes together and we have assembled them and put them, lined them up, and it forms 1 to 3 billion. And they're all numbered 1, 2, 3, 4, 5, 6 because it's all discrete, right? These positions. Let's look at position number 117 in all of us. I may have a C, you may have a T, he may have a G. So different people can have different kinds of nucleotides. But in most positions, if you look at the adjacent position 116, all of us probably have A. If you look at position 117, some of us have C, some of us have T. That means that position number 116 is not variable among individuals. Position number 116 is called monomorphic. One single nucleotide, that's monomorphic. Because position number 117, there is variability amongst us, variability in the nucleotide, that's called a polymorphic position. Poly means more than one, so it's more than one, so it's called a polymorphic position. And of course, you and I, if that particular polymorph 117 position determines our eye color, then you may have a black eye color and I may have a brown eye color because you have a G in that position, I have a T in that position. Right? So have you understood what is meant by a polymorphic position?
Yes.
And that polymorphism can actually impact on our phenotypes. Let's say that determines that gene on which this polymorphic site is located determines eye color, and your having G at that particular position gives you a black eye color. My having a T at that position gives me a brown eye color.
Thank you, sir.
Magic from University. Sir, my question is that can I speak language?
Yes. বাংলা হিন্দি আপনি বলো।
ইন্ডিয়াতে ওরাল ক্যান্সার যেটা হচ্ছে মানে ইন্ডিয়া ওটা নিয়ে প্রোগ্রেস করেছে, অন্য ক্যান্সারগুলো নিয়ে কেন করলো
না? করেছে অন্য ক্যান্সারগুলো নিয়ে করলো না, ইজ নট রাইট। কিন্তু যে ম্যাসিভ স্কেলে ওরাল ক্যান্সার নিয়ে করেছে সেই ম্যাসিভ স্কেলে অন্য ক্যান্সারগুলো নিয়ে করেছে। তার মেজর কারণ হচ্ছে অ্যাকচুয়ালি আমরা দুটো ক্যান্সার নিয়ে করতে চেয়েছিলাম, কিন্তু এগুলো এত এক্সপেন্সিভ, এত কোটি টাকা লাগে যে গভর্নমেন্ট অফ ইন্ডিয়া আমাদের অত কোটি টাকা দিল না। Oral cancer is the most prominent, most prevalent, most frequent type of cancer among males in India, and among females it's cervical cancer. So why is it that government of India funded us for doing oral cancer and not cervical cancer? The reason being that cervical cancer is more homogeneous throughout the world. It's caused by a virus called the human papilloma virus. And আমরা যখন এই প্রজেক্টটা আরম্ভ করেছিলাম আন্ডার দ্য ইন্টারন্যাশনাল ক্যান্সার জিনোম কনসর্টিয়াম, অলরেডি ইউএসএ তে একটা প্রজেক্ট চলছিল যেটার নাম হচ্ছে দ্য ক্যান্সার জিনোম অ্যাটলাস প্রজেক্ট। সেখানে ওরা অলরেডি they had generated massive amounts of data on cervical cancer. They had also generated data on oral cancer, কিন্তু ইউএসএ তে oral cancer is mostly tongue cancer. আর আমাদের এখানে oral cancer is mostly outside of the tongue in the buccal cavity. তার কারণ হচ্ছে যে আমাদের এখানে প্রচুর খৈনি খায় লোকেরা, সেই খৈনিগুলো গিয়ে বাকাল ক্যাভিটিতে, ওই চুনটা গিয়ে বাকাল ক্যাভিটির এপিথেলিয়াম ইরোড করে, এবং তারপরে ওই যে তামাক যেটা থাকে সেটা গিয়ে ডিরেক্টলি সেলগুলো থেকে ইউএসএ তে অত খৈনি কেউ খায় না, রাইট? সো এই কালচারাল প্যাটার্নস এই কারণের জন্যে ইন্ডিয়াতে ওই বাকাল ক্যাভিটি ক্যান্সারস আর মাচ মোর ফ্রিকুয়েন্ট দ্যান টাং ক্যান্সার, ইউএসএ তে ঠিক তার উল্টো। সুতরাং আমরা যখন আরম্ভ করলাম, দ্যাট ওয়াজ ওয়ান আর্গুমেন্ট যে আমরা যদি এখানে ওরাল ক্যান্সারে কাজ করি, ইভেন দো ইউএসএ তে ওরাল ক্যান্সারের কাজ হচ্ছে, ওরা প্রাইমারিলি টাং ক্যান্সারে ডেটা জেনারেট করবে। সো উই ডিসাইডেড দ্যাট উই উইল লুক অ্যাট দি ওরাল ক্যাভিটি উইদাউট দ্য টাং। It's called gingivobuccal oral cancer when you are excluding the tongue. So that's the reason why we looked at oral cancer and not any other cancer type. প্রত্যেকটা ক্যান্সার টাইপ এই ধরনের টেকনোলজিতে আমাদের স্যাম্পল সাইজ ছিল 500। সেই তারও একটা কারণ আছে, সেটা আমি ইলাবোরেট করছি না। 500 মানে হচ্ছে যে অ্যাকচুয়ালি 1000, কারণ আমাদের টিউমার থেকে টিস্যু নিতে হয় এবং নর্মাল থেকে টিস্যু নিতে হয়, কারণ আমাদের একটা কম্পেয়ার করতে হবে তো যে সোম্যাটিক মিউটেশনগুলো কোথায় হচ্ছে। সুতরাং 500 পেশেন্ট নিলে 1000টা ডিএনএ সিকোয়েন্স হোল জিনোম সিকোয়েন্সিং করতে হতো। That cost us about, I would say about 150 crores of rupees. এবং তার থেকে বেশি গভর্নমেন্ট অফ ইন্ডিয়ার ইনভেস্ট করার ওয়েরকাল ছিল না। সুতরাং এই প্রজেক্টগুলো খুব এক্সপেন্সিভ প্রজেক্ট, in spite of the fact the cost of sequencing has fallen. Have I answered your question?
Yes.
Thank you. The question, what is big data? And essentially, the reason why I'm asking this question is that, as I told you, that we are taking one particular DNA fragment randomly, DNA segment, randomly fragmenting it and sequencing these, randomly fragmenting it multiple times, not once, multiple times. Again, you'll be introduced to what's called depth of sequencing. So usually when we look at these kinds of technologies, we take the same DNA piece, do it 30 times, that's called 30x. Because after all, when you're randomly fragmenting it, you may miss out on certain pieces, and also I told you that small pieces we are throwing out. So in one fragmentation you can get one set of small pieces, in another fragmentation you can get another set of small pieces. So after we have put all of these together, start sequencing, we are generating many gigabytes of data. And so this is what's called big data, and to be able to first of all, to be able to store this data becomes a big problem. It requires huge storage, computer storage spaces to be able to do that. Now, even if you have stored the data, why are you storing the data? Because you want to analyze it, right? You need to assemble it, you need to find overlaps and so on and so forth. You need to analyze that data. For analysis of that data, and you want to analyze it in a parallel fashion. To be able to do that, to be able to analyze the data in a parallel fashion, you would need big computing machines, big servers, computing machines, etc. So big data means that you need big infrastructure, big computing infrastructure. But what is big data? What kind of data? So a collection of data sets, which is large and complex. I'm just trying to define big data. So it's so large and so complex that currently available tools, it's difficult to store the data because it's really large. Efficiently manage the data, so you also need to manage the data to be able to, assemble and so on and so forth. Easily access, search and share the data. Oftentimes for parallelization you need to share the data on multiple servers. The servers may not even be located on the same floor in your own institution. The servers can be located in two different institutions to be able to share the data. And then you need to visualize the data. After having assembled, you have the reference sequence and you want to know, visually see how much of overlap is there between the reference sequence and your data. And ultimately, like I said, to be able to analyze the data for drawing useful inferences within the time periods relevant to biological research and translation. So for example, if a cancer patient has come to you and you are asking what kind of somatic mutations resulted in cancer in this one patient, you can't take the tumor tissue and the normal tissue and say, well, go home, come back after 2 years because I'm going to generate the data and analyze the data and then tell you. By the time the patient comes back, the patient is dead. Right? So you can't take 2 years. The doctor will not allow you to take that kind of money. The doctor pressurizes us to generate the data, to analyze the data in rapid time, to draw inferences in rapid time. So we need to draw inferences within time periods that are relevant to biological research and translation. The patient is waiting to know, and the doctor, depending on what kind of somatic mutations this patient has, will determine what kind of treatment the doctor is going to use. Therefore, the doctor cannot wait, the patient cannot wait. So you need to do this parallelly, you need to do this rapidly, but the data are big, it's big data, so how do you do that? Handling of big data becomes a big problem, or handling of data generated by a next generation sequencer has become a big problem because infrastructure that's required, algorithms that are required are still being developed and generated. So you have volume, which is the scale of the data, and you have veracity, which is uncertainty of the data. So you need to draw inferences reasonable, that are correct, mostly correct. So there are four attributes of all of this: volume, veracity. I'll skip this. And then there are two others, which is called variety. So you can generate DNA sequence data, you can generate RNA sequence data, and various kinds of data, epigenetic data, methylation data, and so on and so forth. So there is a variety of data, and the rapidity with which you need to analyze this data is called velocity. So when you have generated big data, there are four V's associated with the big data. One is volume, that is big data. The other is veracity, how efficiently you can, or correctly you can infer about the data. Then the variety is different kinds of data. Like I said, from the same DNA sequence, you can look at, from the same tissue, you can look at DNA sequence, RNA sequence, methylation profiles, and so on. That's different forms of that same data. And velocity, you need to analyze these data rapidly to be able to make your inferences useful. And so these are the four cardinal features of big data. How much of increase in size have there been in terms of data? So this is increase in data size per individual. So if you look at genetic data, which is essentially means that we are leaving out all of the non-coding portions of the DNA. The non-coding portion of the DNA is much greater than the coding portion of the DNA. That's about 2.5 kb. Now, genetic data means that we are looking at candidate genes. We are looking at a small set of genes. So we used to, when I started my life, I would generate data of this magnitude, about 2.5 kb. Then when Sanger sequencers, and at that time you wouldn't even know those technologies, how we used to sequence. We had very large glass plates and so on and so forth. So that's all a part of genomic history. You may not even know any of that history. That's how I started my life. That's where we were generating data of that magnitude. Then came Sanger sequencers, when we were able to generate genome-wide data, very slowly, but still genome-wide. Then it became 100-fold from 2.5 kb to 2 GB. And now when we generate genome-wide data, we can generate genome-wide data on 5 million loci using these chips, etc. So about 50 markers, genetic data, to genome-wide data, about 5 million markers. And here we were generating data on about tens of individuals, and now we generate data on thousands of individuals. Like I said, in the Human Genome Project, in the cancer, oral cancer project, in the Cancer Consortium project, we had 500 individuals, but that's equivalent to 1,000 tissues. So we actually generate a large amount of data. And in the 1990s onwards, we've been generating data on GB scale, not TB scale. So till 1990s in TB scale, 1990 onwards in GB scale, 2005 onwards, when these massively parallel sequencers came, we generate data in terabyte scale. So kilobases or kilobytes, however you want to look at it, kilobytes to gigabytes to terabytes. That's been the evolution of data generation, and these are multiple orders of magnitude larger than the previous. I'm going to skip all of this. I think I'm just going to skip all of this. No, I just want to. All right. So this is one point that I wish to make. I wasn't sure what the audience was going to look like, right? I imagined that they could either be biologists or be information scientists, but it seems like a mix. So I had in my deck of slides both kinds of things. Depending on what the nature of the audience was, I would emphasize one part. So I'll skip all of the mathematically more technical part. So the question that oftentimes arises is that, well, you've gone big. Genome scientists have gone big. You have spent a lot of taxpayers' money. New machines have been invented. Did we gain anything at all? Well, one gain from genomics, at least human genomics, would be our ability to be able to understand how diseases are caused, right? That's where we believe that a lot of diseases are caused by genomic alterations, and therefore you should be able to identify genomic alterations that relate to certain specific kinds of diseases. So this is a map that actually shows you the positions of disease-causing markers, disease-causing loci on different chromosomes. So these are the different chromosomes in the human genome, and each dot there represents a disease. So what I want to impress upon you is that by going big, we have actually been able to map a very large number of diseases on the chromosomes, on specific regions of the chromosomes. For most of them, we now know the genes. For most of them, we know what these genomic alterations do to the genes. Functionalization of the genes have also been done, right? So going big really provided us with a lot of empowerment of understanding these diseases, and if you don't understand a disease, you can't get a treatment or even manage the disease well. So right now, because of all of this, improvements in genomics platforms, next generation sequencing, ability to generate, ability to analyze data, we have actually gained a lot. And I'm only talking about humans. Same thing with agriculture, same thing with animal genetics, milk production, and so on and so forth. So lots of meat, there's a huge increase in the consumption of meat. Now you need to the quantity of meat in an animal is actually genetically controlled. So can you do certain kinds of interesting things to be able to increase the amount of meat on a specific animal? So increase the production of rice by increasing the amount of rice grains on a single rice plant. This is all done by understanding what genes are involved in the production of rice and so on and so forth. So I'm only talking about the human genome, and the answer is that we did gain a lot of knowledge, and therefore did gain a lot of practical ability to control diseases by going big. Similarly, in other genomes you can think of, in other species you can think of these kinds of things. I think I should stop. I've been talking about this. This is another entirely different set, which is again interesting. That the big data, the next generation sequencing data, have resulted in a huge amount of collaboration between scientists of different domains. I have talked about biology. I've talked about information science. And this has also resulted in a huge collaboration between statistical sciences and computer sciences. The next set of slides pertains to that, where statistics meets computer science, and it meets on the field of next generation sequencing data. Again, the reason why these kinds of hybrid or crosstalk is required is because, as I said, you need more efficient algorithms, you need faster algorithms to be able to make an impact on human health, to be able to, that patient who is waiting to figure out why I've gotten this disease, and the doctor wants to know how I can treat that disease, you need to rapidly come up with a diagnosis of all of this. So these are the reasons why these different domains of science have started to interact, and they have come up with very, very useful methods, methods that are very useful for rapid inferences from next generation sequencing data. So I will end my talk by saying that the title of my talk was what is it that why did we need next generation sequencing? What kind of application? So I started with that. I started with one example of cancer, and I've shown you that I hope that I've shown you that without next generation sequencing platform, we wouldn't even be able to ask the questions that we really want to ask. And now that we can ask these questions, we generate data, we need algorithms to be able to analyze the data, and this comes in the area of bioinformatics, and that's what this workshop is all about. So this workshop will start with where the wet lab generation of data ends. This workshop will begin from there, start with the data and tell you about the various kinds of algorithms. I have not touched, like I said, the protein modeling and so on, but that will also be a part of this particular workshop, and I sincerely hope that you gain a lot from this workshop. Even though it's not hands-on, there will be demos, there will be demonstrations of the methods, but like I said, that I've encouraged Professor Bakshi, and I also have some impact on the National Institute of Biomedical Genomics. Maybe the two institutions should come together and do a one-week workshop and bring all of you back so that you can do hands-on work on those methods and data sets, real data sets. Thank you very much for your attention.
Though it is a keynote lecture, but sir, as sir mentioned, that it is a traditional keynote lecture. So I expect a couple of questions from the audience. Yes, go ahead.
Good afternoon, sir. By finding any kind of maybe drawbacks in a particular technique can help us to improvise the technique even more.
Oh, always.
So by doing it, we came across certain advantages that we didn't have. Is there any drawback or problem that we are facing by doing it?
Oh, absolutely. We don't even have the space to store the data. We generate data, the next generation sequencer will generate the data, and it will tell you that the sequencer will say that take the data, keep it somewhere, analyze the data. You don't even have the space to store the data. You don't have the servers to analyze the data. So those are the immediate fallouts of generating big data. After that, there are a multitude of issues that we have faced in terms of improving the algorithms. There are 4 V's. Those 4 V's are pertain to big data, and each of those 4 V's actually we have stumbled on various kinds of known algorithms, and then we've tried to improve it. Improvements are going on even today. The algorithms that we use today were not the algorithms that we were using even 6 months ago. That rapid pace these are changing. The algorithms are changing. Yeah.
And we expect sir to come here again in the next workshop, because we conduct such workshops in near future again. So this time now I'm going to declare it as a lunch break. We will be back here at 2. And from 2 onwards...
Hey, you can't be back here at 2.
Oh, it's only, we have 15
1:45.
Okay, so 2:15 sure. So, and from then onwards, we will go to the hands-on demonstration. So, try to be back within 2:15.
Thank you very much.